Hey guys, it's Jacob here again. Today is May 25th, 2019, and today we're going to be going over our microscopic compound and everything that our microbiology really revolves around, the microscope. Alright, so let's go ahead and get started. I'm going to name off a few of the areas of the microscope here. So you have your ocular lens right here at the top. It's what you see through to look down at your specimen on the slide. You have your arm right here. And then you have your turrets of the microscope. I think they call it a turret because it kind of like spins around like a turret gun. It's kind of cool actually. And then you have your objective lens right here, the big red one. And then you have your condenser lens right here, which is a blue one. Alright. Making our way down, we have our we have our coarse focus right here, which allows you to focus whatever specimen you have right here in a vertical way. And you have your fine focus, which is more horizontal from what I've seen so far in our discoveries. And then you have, of course, your stage. This is what you put the actual slide on. And also you have your stage clamp. So you just pull this back like that. Put the slide right there, and it closes perfectly just like that. Alright. And then right here you would have your illuminator, which is going to light up the specimen right here on the slide. And then you have your base at the bottom right here, which allows the microscope to stand up on its own. Alright, and then lastly you have your diaphragm right here, which is kind of like, I guess, a separate middle part of the microscope. So now I'm going to be talking about how to make your own slides. So pretty much you get the slide. So say for example I'm going to swab my cheek like we did. So I swab my cheek with the swab. I rub it on the slide like so, like that, and then I get some condensed water, not condensed water, what is it called? Distilled water. Distilled water, you take a drop of that, you drop it on there, and then you take your slide cover, you want to tip it just like that, and then drop it. Alright, anything else, any like other excess water you kind of want to dry up with like a towel without touching the specimen. And then you just simply pull the clamp back, put it on there, clamp it. And then, so this microscope doesn't have a light on it, so we used a phone light and put it underneath. And then you just look through it like this from the ocular lens, and then you adjust it accordingly to how you want. You know, you have left, right, up, down. So yeah, it's pretty simple. It's pretty fun. So that's the microscope. You really want to know your microscope and how to use it before you really dive into looking through it and getting into the experiment stuff. All right. So that was the microscopic video this week for knowing your compound. I hope you enjoyed it, and I'll see you on the next one. Thank you.